When we picture how the brain communicates, I think most of us fall back on a pretty simple metaphor.
Right.
We imagine it a bit like a post office. You think of a neurotransmitter, say serotonin or dopamine, as a letter. One neuron seals the envelope, slides it across the counter of the synapse, the receiving neuron picks it up, and boom, message delivered. We tend to view the synapse as the whole story.
It is a very clean sort of binary way of looking at it. And it's really how a lot of early neuroscience was taught.
Yeah.
The assumption was that the letter is either delivered or it isn't, and the receiving cell simply acts on the instructions straight away.
Right. But then you start looking into the actual mechanics of intraneuronal signaling. And you quickly realize that handing the envelope over the counter is, well, it's literally just the preamble. The real heavy lifting, the opening of the letter, the decoding of the message, cross-referencing it with other incoming mail, deciding how to respond, and potentially rewriting the entire branch's rule book based on that one letter, all of that happens inside. It happens in this massive, incomprehensibly complex sorting room inside the receiving neuron.
Which is the absolute definition of a complex dynamic system. And this actually brings us to a core philosophy that we really need to anchor ourselves to for this deep dive.
Okay, let's hear it.
Psychiatry at its heart is the study of how brain, mind, and experience interact. But more importantly for us as clinicians, it is the study of how that precise interaction can go wrong. If we only look at the synapse, the empty space between the cells, we are completely missing the actual decision-making apparatus of the brain.
We're missing the interpreter.
Spot on.
And that's exactly what we are focusing on today. We're diving into some fascinating material, pulling heavily from the foundational texts of psychiatry, specifically Chapter 1.11 of Kaplan and Sadok's Comprehensive Textbook of Psychiatry.
It's a dense chapter, but a crucial one.
Very dense. But our mission here isn't just to list out a bunch of disconnected biochemical pathways, right? We want to move from the microscopic mechanism to the clinical manifestation and ultimately to the clinical meaning. We need to figure out how neurons don't just passively receive signals, but actively interpret them.
It represents a profound shift in perspective. I mean, the neurotransmitter is merely the first messenger. What happens next, deep within the cytoplasm and the nucleus, dictates everything from how a childhood memory is permanently stored to why an antidepressant takes 4 weeks to show any therapeutic effect.
Yeah, that's such a massive point. Now, to you, the listener, I know that whenever we start throwing around terms like kinases, phosphates, and intracellular cascades, it can trigger a bit of medical school PTSD.
Oh, definitely. It's a lot of molecular alphabet soup.
Right, we're going to get into the biochemical weeds today, but do not let that overwhelm you. Our goal is to build robust mental models. We want to understand the why and the how behind these systems, not just memorize the what. Because if we can visualize that sorting room, your clinical reasoning becomes so much stronger.
And building that mental model really starts with grasping the overarching concept of signal transduction. At its most fundamental level, signal transduction is the process by which a cell converts an extracellular signal, like our neurotransmitter floating in the synaptic cleft, into an intracellular cascade.
Okay.
So the ligand binds to a receptor on the plasma membrane. That binding actually stabilizes a unique three-dimensional physical shape of the receptor, which then transmits the signal across the fatty cell membrane to a network of adapter proteins or enzymes waiting on the inside.
Okay, let's unpack this right away because this is where my brain always hits a roadblock.
Yeah.
Why don't I just have a direct wire?
A direct wire.
Yeah. Why do neurons need a multi-step Rube Goldberg machine of proteins, second messengers, and kinases just to pass a message? Think about it. If the receptor on the outside has already caught the neurotransmitter, the cell already knows the message is there.
Right. It seems redundant.
Exactly. Why go through 10 different highly volatile biochemical steps just to open an ion channel or turn on a gene? It seems incredibly inefficient to me.
It looks inefficient until you consider the alternative, really. A direct wire is actually far too rigid for a biological system. Evolution settled on these multi-step cascades because they offer 3 massive survival advantages: amplification, integration, and temporal or spatial control.
Okay, let's take those one by one. Amplification. How does the Rube Goldberg machine act as an amplifier?
Think about the physical limitations of a direct wire. If one signal on the outside strictly equals one action on the inside, the cell is entirely dependent on the volume of the incoming neurotransmitter.
So 1-to-1 ratio.
Exactly. But with signal transduction, one single neurotransmitter molecule binding to a receptor can activate an enzyme. That single enzyme can produce hundreds or thousands of second messenger molecules.
Oh, wow.
And it doesn't stop there. Each of those second messengers can activate a kinase, and each kinase can phosphorylate thousands of downstream targets.
So it's like a single person whispering into a microphone, but that microphone is hooked up to a stadium-sized speaker system.
That is a brilliant analogy, yes. You allow the cell to have a massive, highly sensitive response to a very faint stimulus.
That makes perfect sense. Okay, what about integration?
Integration is about decision-making. Neurons are rarely just listening to one signal. I mean, they are constantly bombarded by excitatory signals, inhibitory signals, hormones, inflammatory markers, all at the exact same time.
Just a constant barrage of mail.
Right. If they had direct wires, it would just be noise. But these multi-step pathways allow the cell to maintain separate channels of information that can physically crosstalk inside the cytoplasm.
So they act kind of like logic gates in a computer.
Precisely like a biochemical A&D gate. A neuron might be programmed so that it only fires or only changes its synaptic structure if it receives signal A and signal B simultaneously.
Right, so the intracellular cascades physically intersect to calculate that coincidence.
Exactly. The sorting room decides that signal A alone is meaningless, but signal A plus signal B means we need to build a new memory.
That is fascinating. It's not just a relay race, it is a live computational process.
Yeah.
And the third reason was temporal and spatial control. How does the cascade control time and space?
That really comes down to context. These complex pathways allow the neuron to tune its own sensitivity based on what's happening right now and, crucially, what has happened in the past.
Okay.
It is highly advantageous for a neuron to be incredibly sensitive to a rare, novel stimulus, but to completely ignore a repetitive, annoying one. The intracellular machinery has built-in feedback loops that decide to turn the volume up or down. It determines a neuron's responsiveness based on its history.
If you've ever wondered why your morning coffee doesn't give you the same jolt it did a year ago, you are experiencing that temporal control firsthand. Your sorting room has actively downregulated its response to the incoming mail.
Quite right.
Now, to structure our exploration of this sorting room, the source outlines 3 main schemes or receptor types that handle this signaling. Let's briefly map them out before we dissect them.
We can categorize them broadly by speed and function. First, we have ligand-gated ion channels. These mediate fast synaptic transmission acting in milliseconds. This is primarily for your amino acid neurotransmitters like glutamate for excitation and GABA for inhibition.
That's the fast lane.
Then second, we have G-protein coupled receptors, or GPCRs. These mediate slow synaptic transmission taking seconds to minutes, and they rely heavily on those second messengers we mentioned.
Okay.
Finally, the third scheme involves receptor tyrosine kinases, or RTKs. These are primarily used by neurotrophic factors, growth factors, basically, and they trigger complex kinase cascades to regulate structural growth and gene expression over hours or days.
Fast, slow, and growth. Let's bypass the fast ion channels for a moment and start with the slow but incredibly powerful sorting machinery. The G-protein switch and second messengers. We are talking about GPCRs, and we really need to emphasize just how ubiquitous these are.
Oh, absolutely everywhere.
GPCRs account for at least 2% of the entire human genome. They are the primary targets for a massive array of molecules we care about in psychiatry, serotonin, dopamine, or epinephrine. If you prescribe a psychiatric medication, odds are you are interacting with a GPCR.
They are the absolute workhorses of psychopharmacology. Visually, a GPCR is a single long protein chain that snakes back and forth across the fatty cell membrane exactly 7 times. Because of this, they're sometimes called 7 transmembrane receptors.
7 times. That's very specific.
It is. But the real magic is on the inside. Hanging off the bottom of this receptor, inside the cell cytoplasm, it is physically associated with a heterotrimeric G protein.
Heterotrimeric, meaning 3 different parts.
Yes. An alpha subunit, a beta subunit, and a gamma subunit, all clustered together.
Okay, so we have our receptor weaving through the membrane, and docked underneath it is this 3-part G protein. Now, walk me through the physics of the actual switch. How does a chemical hitting the outside turn this thing on?
It revolves around a tiny nucleotide called guanosine diphosphate, or GDP. In the resting state, when nothing is happening, the alpha subunit of the G protein is tightly clutching a molecule of GDP. As long as it holds GDP, the whole 3-part complex is inactive. It's locked.
Got it.
But when a neurotransmitter, let's use dopamine as our example, binds to the extracellular pocket of the GPCR, it alters the electrical charge of the receptor.
And because proteins are just folded chains of amino acids with different charges, changing the charge changes the physical shape of the entire receptor.
Exactly. That physical shift twists the intracellular tail of the receptor. This twist acts like a lever, physically forcing the alpha subunit to open its binding pocket and drop the GDP molecule.
It literally pries the GDP out of its hands.
Yeah.
What takes its place?
Well, nature abhors a vacuum. Floating abundantly in the cytoplasm is a very similar molecule, but with a higher energy state. Guanosine triphosphate, or GTP. The moment the alpha subunit is empty, a molecule of GTP slips in and binds to it. When GTP binds, the molecular switch is officially flipped to on-N.
And what does on look like physically?
The binding of GTP causes another massive shape change. The alpha subunit physically detaches from the receptor, and it also breaks apart from the beta-gamma complex.
So they separate.
Yes. You now have two distinct active entities, an active alpha subunit and an active beta-gamma dimer. They are now free to slide along the inside of the cell membrane and interact with other enzymes to do the actual work.
They break apart and go their separate ways to wreak biochemical havoc.
Yeah.
But here's a biological paradox. If they break apart and float away, how does the sorting room ever shut them down?
That's the million-dollar question.
Right, because if they just stayed active forever, the cell would never stop firing. You'd have runaway signaling, which in the brain usually manifests as a seizure or excitotoxicity.
That is where the elegance of the G protein really shines. It has a built-in countdown timer. The alpha subunit isn't just a messenger, it is an enzyme itself. It has intrinsic GT-PACE activity.
Meaning what exactly?
This means the alpha subunit slowly hydrolyzes or breaks down the GTP molecule it is holding, clipping off one phosphate to turn it back into GDP.
Wait, it turns itself off?
Yes, precisely. The moment that GTP is clipped back into GDP, the alpha subunit instantly reverts to its inactive shape. It stops interacting with its targets, turns around, and reassociates with the beta-gamma complex and the receptor. The entire system returns to its quiet basal state, ready for the next dopamine molecule.
That is incredible, a self-timing switch. So while they're active and roaming around the membrane, what exactly are they doing? The source material highlights two major second messenger pathways triggered by these G proteins. Let's start with the absolute classic, the cyclic AMP or CAMP pathway.
The CAMP pathway is arguably the prototypical second messenger system. It is heavily regulated by two specific opposing classes of G proteins, G stimulatory, which we call Gs, and G inhibitory, which we call G.
So it's a built-in tug of war.
Exactly. When a receptor coupled to Gs is activated, say a dopamine D1 receptor, its active alpha subunit seeks out a large membrane-bound enzyme called adenylate cyclase. It binds to it and stimulates it.
Okay.
Adenylate cyclase then starts grabbing ATP, the main energy currency of the cell, and rapidly converts it into cyclic AMP.
And if a neurotransmitter hits a G-coupled receptor, like a dopamine D2 receptor,
then the G-alpha subunit floats over to that exact same adenylate cyclase enzyme and inhibits it, slamming the brakes on CAMP production. So the net level of CAMP in the sorting room at any given millisecond is the mathematical sum of the G's gas pedal and the G brake.
Okay, so let's say the gas pedal wins. We have a massive spike of CAMP molecules now floating in the cytoplasm. What is their actual job?
Their primary mission is to find and activate a specific protein called protein kinase A, or PKA. Now, as a quick refresher, a kinase is an enzyme that takes a phosphate group from ATP and slaps it onto another protein.
And adding a phosphate changes things.
Tremendously. Adding a highly negatively charged phosphate group changes the shape and therefore the function of that target protein. It can open an ion channel, activate another enzyme, or move a vesicle.
But PKA isn't always active, right? If it was, the cell would be in constant chaos.
No, at baseline, PKA is inactive. And the mechanism is quite clever. PKA consists of 4 parts, 2 regulatory subunits and 2 catalytic subunits. The regulatory subunits physically wrap around the catalytic subunits, covering up their active sites.
Like a safety mechanism.
Exactly. It's like having a sharp pair of scissors, but with a thick plastic safety cover locked over the blades.
And CAMP is the key to the safety cover.
You've got it. It takes 4 molecules of CMP to bind to the regulatory subunits. When they bind, the regulatory subunits change shape and let go. The catalytic subunits, the sharp scissors, are now free. They detach and go roaming through the cell, phosphorylating their targets.
Here is where this gets vitally important for psychiatric practice. These catalytic subunits of pKa don't just hang out near the membrane tweaking ion channels. They can actually travel deep into the cell. They can enter the nucleus.
Yes, and this crosses a critical threshold. When pKa enters the nucleus, it phosphorylates a specific transcription factor called CRA, that's CMP response element binding protein.
A transcription factor. So we're talking about interacting directly with DNA now.
We are. Once CREB is phosphorylated by pKa, it becomes active. It binds to specific promoter sequences on the DNA and recruits the cellular machinery to either increase or decrease the transcription of target genes into messenger RNA.
Let's pause and really let the clinical meaning of this sink in. A patient swallows a pill, say, an SSRI. Within hours, the serotonin levels in their synaptic clefts rise. The serotonin binds to the GPCR. The G protein flips. Adenylate cyclase makes KMP. KMP unleashes pKa. pKa travels into the nucleus and activates CRE.
Exactly.
And CREB starts reading the DNA to build entirely new proteins, new receptors, new structural supports for the neuron. This is the molecular answer to the most common question in psychiatry. Why does my antidepressant take 4 to 6 weeks to work if it raises serotonin on day 1?
It's such a crucial point for trainees to understand.
We aren't just waiting for a neurotransmitter to float across a gap. We are waiting for the neurons to transcribe new genes, manufacture new proteins, ship them down the axon, and physically rebuild the architecture of the brain. The short-term chemical signal has been interpreted and translated into a long-term structural adaptation.
It perfectly illustrates why the synapse is just the beginning of the story. You are fundamentally altering the hardware of the brain, not just its software. But as with all biological pathways, if you turn it on, you must have a way to turn it off. How do we clear the KMP once the signal has passed? Because if KMP stays high, pKa stays active, and CREB keeps transcribing.
The sorting room has to be swept clean.
It is swept clean by a family of enzymes called phosphodiesterase, or PDEs. Their entire job is to hunt down KMP, cleave a specific chemical bond, and turn it back into regular, inactive AMP. This effectively shuts down the entire pKa pathway.
And the source points out something brilliant here. If we know that PDEs destroy CAMP-AP, then a drug that blocks PDEs would artificially keep CAMP levels high, prolonging that active, excitatory state of the neuron. And millions of people self-medicate with a non-selective PDE inhibitor every single morning. Caffeine.
Exactly. Caffeine antagonizes adenosine receptors, but it also inhibits phosphodiesterases. By blocking the cleanup crew, caffeine ensures that whatever CMP is being produced stays around longer, amplifying the wakefulness and excitatory signals in the brain.
And the precision of our pharmacological tools is getting even better. The text notes that sildenafil Viagra doesn't just block any PDE. It targets a very specific isoform, PDE5, which is heavily concentrated in vascular smooth muscle.
Right.
Doesn't affect the brain much, but it massively alters the intracellular signaling in blood vessels. It's a perfect example of how targeting these intracellular termination steps deep in the sorting room opens up entirely new classes of therapeutics.
It proves that the future of drug development lies beyond the cell's surface. Which brings us to the second major G protein pathway outlined in the text, the phosphatidylenosytol, or PI pathway. We talked about G's and G. This pathway is driven by the GQ class of G protein.
But GQ pathway. Okay, in the CAMP pathway, our target enzyme was adenylate cyclase.
Right.
What enzyme does the GQ alpha subunit hunt down?
The active GQ alpha subunit activates a different membrane-bound effector enzyme called phospholipase, or PLC.
Phospholipase, so it cleaves a lipid, it cuts a fat.
Yes. The inner leaflet of the cell membrane is packed with various phospholipids. PLC specifically looks for a fat molecule called PIP2. That's phosphatidylenosytol 4-gol-5-bisphosphate.
Okay, PIP2.
When PLC finds PIP2, it acts like a pair of molecular scissors and cuts it right down the middle, generating two distinct second messengers, DAG, which is diacylglycerol, and IP3, inositol trisphosphate.
So one starting material gets chopped into two separate tools, and because of their chemistry, they behave very differently. DAG is a fat, so it hates water. It stays embedded in the fatty cell membrane. But IP3 is heavily phosphorylated. It's highly water-soluble, so it immediately detaches from the membrane and floats away into the watery cytoplasm. Let's track IP3 first. Where is it swimming to?
IP3 diffuses deep into the cell until it encounters the endoplasmic reticulum. Now, normally we think of the ER as the place where proteins are folded, but it serves another vital function. It is an immense, tightly sealed storage vault for calcium ions. The concentration of calcium inside the ER is thousands of times higher than in the surrounding cytoplasm.
Which means if you open a door, The calcium isn't just going to drift out. It is going to explode out down its concentration gradient.
Exactly. And IP3 is the physical key to that door. IP3 binds to specific ligand-gated channels on the surface of the endoplasmic reticulum. The channels spring open, and a massive flood of stored calcium rushes into the cytoplasm.
Now, I need to pause here, because calcium is a tricky element. Why is the cell locking it in a vault in the first place? Why go through all this trouble just to release a mineral?
Because free calcium in the cytoplasm is incredibly dangerous, but also incredibly powerful. If calcium levels stay high for too long, it triggers apoptosis-programmed cell death. The cell literally destroys itself. But in short, controlled bursts. Calcium is one of the most potent second messengers in biology.
So the IP3 triggers the burst. What does the calcium actually do once it's free?
The calcium ions immediately seek out and bind to a specialized, ubiquitous protein called calmodulin. You can think of calmodulin as a molecular claw. Once 4 calcium ions bind to it, calmodulin undergoes a massive conformational change, becomes active, and goes on to wrap around and activate an entire family of CAM kinases.
Okay, another kinase cascade.
Exactly. These CAM kinases then proceed to phosphorylate multiple downstream targets, much like pKa did in the other pathway.
So we have a massive calcium cascade triggered by IP3. But we can't forget the other half of the scissors cut. While IP3 is opening the calcium vault deep in the cell, what is DAG doing back at the membrane?
DAG remains anchored in the plasma membrane and acts as a chemical beacon. It recruits an enzyme called protein kinase C, or PKC, pulling it out of the cytoplasm and up to the membrane. Interestingly, many isoforms of PKC also require the calcium released by IP3 to bind to them before they become fully active.
Oh, look at the integration there. The two halves of the cut PIP2 molecule, DAG and IP3, have to cooperate. IP3 releases the calcium, which travels up to the membrane to help DAG fully activate PKC. It is a beautifully coordinated, two-pronged attack from a single starting molecule.
It is the essence of biological redundancy and integration. But, just like the CAMP pathway, we have to ask, how does the sorting room clean up this mess? The IP3 has to be cleared, and more importantly, it has to be recycled, because the cell needs a constant supply of PIP2 in the membrane to keep the system running.
The cell is fundamentally frugal. It recycles everything.
Yes, it has to be. So enzymes called phosphatases begin stripping the phosphate groups off of IP3 step by step, converting it back into free inositol. That free inositol is then transported back to the membrane, where lipid kinases use it to rebuild PIP2, restocking the membrane for the next signal.
And here's where we hit one of the most significant clinical connections in the entire textbook. We are talking about lithium. For over half a century, lithium has been the gold standard for treating bipolar disorder, pulling patients out of devastating manias. And for decades, its mechanism was a complete black box. But the source explicitly highlights lithium's action right here in this recycling plant.
It is a remarkable piece of pharmacology. Lithium directly inhibits those inositol phosphatases, the specific enzymes responsible for the final steps of recycling IP3 back into free inositol.
So lithium acts as a roadblock in the recycling plant? What happens to the cell when that roadblock is in place?
You get a traffic jam. Intermediate phosphate molecules start to accumulate, and more critically, the cell experiences a severe depletion of free inositol. Without free inositol, the cell cannot rebuild PIP2.
You're starving the supply chain.
Exactly. This is known as the inositol depletion hypothesis. If a patient is in a manic state, we theorize that certain GPCR pathways, perhaps driven by excess dopamine or glutamate, are wildly overactive, constantly cleaving PIP2 and flooding the cell with calcium and PKC activity.
Wow.
So lithium doesn't block the dopamine receptor. Instead, it quietly throttles the supply chain of the second messengers. As the PIP2 stores dwindle, the overactive signaling pathways gradually lose their fuel and dampen down.
That is such a profound way to conceptualize it.
Yeah.
You aren't boarding up the post office. You're just slowly restricting the amount of paper they have to write the letters on, bringing the chaotic system back into a stable equilibrium. It explains why lithium takes a week or two to work. You literally have to wait for the PIP2 stores to run down.
Spot on.
Before we move away from second messengers, the source touches on two others that often get overlooked: arachidonic acid and gases. Can we demystify those briefly?
Certainly. The arachidonic acid pathway is another lipid-cleaving mechanism. Instead of phospholipase C, various receptors activate phospholipase A2. This enzyme cleaves different membrane phospholipids to release arachidonic acid.
Right.
Arachidonic acid is then rapidly metabolized by two major enzymes, cyclooxygenase, or COX, to form prostaglandins, or lipoxygenase, LOX, to form leukotrienes.
And as soon as you say COX and prostaglandins, any clinician immediately thinks of inflammation and NSAIDs like ibuprofen. Why is this in a psychiatry textbook?
Because the artificial barrier between the immune system and the nervous system has completely collapsed in modern research. We now know that psychological stress induces robust neuroinflammation. The source notes that COX inhibitors are actively being studied as adjunctive treatments for schizophrenia and major depressive disorder.
That's fascinating.
By dampening the prostaglandin second messengers in the brain, we may be able to reduce the stress-mediated inflammatory damage that exacerbates these illnesses.
It's an incredible frontier.
Okay.
And what about the gases? A gas acting as an intracellular messenger sounds counterintuitive.
It forces us to rethink everything we just discussed.
Yeah.
The primary gas is nitric oxide. It is synthesized by an enzyme called nitric oxide synthase, which, fascinatingly, is activated by that exact calcium palmodulin complex we talked about from the IP3 pathway.
Everything is connected. But once the cell manufactures this nitric oxide gas, what does it do? It can't be stored in a vesicle.
That's the key. Because it is a highly volatile lipid-soluble gas, it simply diffuses freely in all directions, passing right through the cell membrane. It completely ignores all the rules of classical synaptic transmission.
Oh, wow.
Often, it acts retrogradely. It diffuses out of the receiving postsynaptic neuron, travels backward across the synapse, and enters the presynaptic neuron that originally sent the signal.
Let's examine the biological logic of that. Why would a receiving cell manufacture a gas just to send a signal backward to the cell that just talked to it?
It acts as a receipt of delivery. It tells the presynaptic neuron, I received your message loud and clear and my intracellular calcium is spiking. Once inside the presynaptic cell, nitric oxide binds to an enzyme called guanylate cyclase, which produces cyclic GMP, a cousin of CAMP.
Right.
This cGMP then alters neurotransmitter release for the next time. It is a critical feedback loop for coordinating the two cells during learning and memory formation.
A chemical read receipt. I love that. It shows how fluid the boundaries between these cells really are.
Yeah.
Now, let's shift our focus entirely. We've spent a lot of time in the slow lane with GPCRs. Now we need to look at the third major scheme outlined in the text, receptor tyrosine kinases, or RTKs.
Yes.
We are moving away from classical fast-acting neurotransmitters and entering the realm of neurotrophic factors, molecules like nerve growth factor, NGF, and brain-derived neurotrophic factor, BDNF.
This is a crucial pivot. We are no longer talking about simply modulating the electrical firing rate of a neuron. Neurotrophic factors operate on a different scale entirely. They dictate massive structural changes, cellular growth, differentiation, robust synaptic plasticity, and fundamentally whether a cell survives or undergoes apoptosis. These factors bind to a specific family of RTKs known as the TRAC receptors.
And the mechanism of action here is completely different from the GPCRs. With a GPCR, you had one receptor snaking through the membrane. But with TRAC receptors, you actually need two separate receptors.
That's right.
When a molecule of BDNF floats up, it acts like a bridge, causing two separate TRAC receptor proteins in the membrane to physically slide together and lock into a pair. The source calls this dimerization.
Dimerization is the ignition switch. Once these two receptors are pulled together, a fascinating reaction occurs on the inside of the cell. The intracellular tails of these receptors possess intrinsic tyrosine kinase activity. When they are forced into close proximity, they actually phosphorylate each other. One tail grabs an ATP and slaps a phosphate onto the tyrosine residue of its partner, and vice versa.
They activate each other. Autophosphorylation.
Exactly. And this autophosphorylation transforms the intracellular tails of the receptors into highly specific, negatively charged docking stations. Suddenly, various adapter proteins that are just floating inertly in the cytoplasm recognize these phosphorylated tyrosines and snap into place, docking at the receptor.
One of those adapter proteins is called GRB2. And I love the analogy of a bureaucratic corporate chain of command for this next sequence. GRB2 docks at the receptor. It doesn't actually do any phosphorylating itself. It's just an adapter. But it recruits another protein from the cytoplasm with the fantastic name SOS, son of sevenless.
It's a very memorable name.
Let's do a quick historical detour because that name is wild. Son of sevenless comes from early genetics research in fruit flies. Scientists found a mutation that caused flies to lack the seventh photoreceptor in their eye, so they called the gene sevenless. When they found a downstream gene that caused the exact same blindness, they logically named it son of sevenless.
Right.
It's a great reminder that these fundamental pathways are conserved across millions of years of evolution.
But back to the sorting room. SOS is basically a middle manager. Its only job is to dock at the membrane and activate a small, critical protein called RAS.
And RAS should sound somewhat familiar conceptually. It is a small G protein. It operates on the exact same GDP and GDP molecular switch we discussed with the massive heterotrimeric G proteins, but it operates solo. SOS acts as an exchange factor. It physically interacts with the RAS, causing it to drop GDP and pick up GTP.
And once RAS picks up GTP and flips to N, it kicks off what I can only describe as a frantic, cascading relay race of kinases, the MAPK cascade. Mitogen-activated protein kinase. It is a three-tiered cascade. Active RAS recruits and activates a kinase called RAF. RAF is a MAPK kinase.
Not harmful.
It is. RAF then phosphorylates and activates MEK. MEK is a MAP kinase. MEK then phosphorylates and activates ERK. ERK is the final MAPK kinase.
It's one manager stamping an approval, handing it to a regional manager who stamps it and hands it to the vice president. And at every single tier, the signal is massively amplified. One RAS activates dozens of RAFs, which activate hundreds of MEKs, which activate thousands of ERKs. And where does ERK take the final stamped message?
ERK travels straight to the CEO. It translocates into the nucleus, where it phosphorylates and activates a host of critical transcription factors, including our old friend CR, as well as others like CRFA. The company policy is officially changed. The cell begins transcribing the genes necessary for robust survival, growth, and the branching of new dendrites.
It is a beautifully brutal, evolutionarily ancient way of passing a tightly controlled, highly amplified signal straight to the DNA. But RAS doesn't only trigger that one relay race. The text highlights that it also activates another massive, clinically vital pathway, the PI3K pathway.
Yes, PI3K phosphoinositide 3 kinase. This pathway takes us back to the membrane lipids. PI3K is a lipid kinase. It takes that same membrane fat we discussed earlier, PIP2, but instead of cutting it in half like phospholipase C did, PI3K adds a third phosphate group to it, creating PIP3.
PIP2 becomes PIP3, and what does PIP3 do?
PIP3 acts as a highly specialized, highly attractive magnetic patch on the inner membrane. It draws a very specific critical kinase out of the cytoplasm. ACT, also known as protein kinase B. Once ACT docks at the PIP3 patch, it becomes fully activated by other kinases.
And ACT is a heavy hitter. The source notes it phosphorylates proteins that actively inhibit apoptosis, essentially ensuring the neuron stays alive under stress. But the real star of the PI3K ACT pathway, at least in the context of current cutting-edge psychiatric research, is a downstream target called mTOR, the mammalian target of rapamycin. The source describes mTOR as a central signaling node, a master controller. Why does mTOR get that title?
Because mTOR solves a massive logistical problem for the neuron. Up until now, we've talked about transcription factors like CERB going to the nucleus to build new proteins, but think about the physical architecture of a neuron. You have the cell body where the nucleus is, and then you have a dendritic tree that can branch out for millimeters or an axon that can stretch for feet.
It's a massive distance on a cellular scale.
Precisely. If a specific synapse at the very distal end of a dendrite experiences a strong burst of learning and needs to rapidly build new receptors to strengthen itself, it cannot afford to send a signal all the way back to the nucleus, wait for mRNA to be transcribed, wait for proteins to be built in the cell body, and then wait days for those proteins to be physically shipped down the dendrite. The memory would be lost.
It needs on-site manufacturing. It needs a local factory.
Exactly. And mTOR is the foreman of that local factory. mTOR regulates the translation machinery, the ribosomes, that are already stationed out in the far reaches of the dendrites. When mTOR is activated by pathways like ACT, it immediately grabs local pre-existing mRNA and facilitates the rapid local synthesis of new proteins. It builds new AMPA receptors, new scaffolding proteins, and new structural supports right there at the synapse within minutes or hours.
Let's tie this directly to the clinic, because the clinical meaning here is revolutionary. The source explicitly connects this mTOR pathway to ketamine.
It's one of the most exciting areas in our field right now.
For decades, we relied on SSRIs, which we established earlier take weeks to work, because they rely on that slow trek to the nucleus via CAM-MP and CRM. But then ketamine emerges as a rapid-acting antidepressant. A patient in a severe treatment-resistant depression or suicidal crisis receives an IV infusion of ketamine, and within a few hours, the depressive symptoms can dramatically lift.
Yes.
The working biological theory, as outlined here, is that ketamine rapidly disinhibits glutamate signaling, which causes a massive immediate spike in BDNF release. That BDNF activates the TRK receptors, which activate ACT which powerfully activates MTR directly in the dendrites of the prefrontal cortex.
It forces the local factories into overdrive.
Exactly. The MTOR triggers a rapid burst of local protein synthesis, generating entirely new dendritic spines within hours. Chronic stress and depression literally wither away the synaptic connections in the prefrontal cortex. Ketamine, via MTOR, physically regrows that withered hardware in real time. It is a complete paradigm shift. We aren't just tweaking the levels of a chemical soup, we are directing the cell's internal master controllers to rebuild the brain's circuitry on demand.
It highlights why understanding the sorting room isn't just academic trivia, it is the very foundation of the next generation of psychiatric treatments. And that leads us perfectly into another pathway that is gaining massive interest for its role as a master controller, want signaling and the GSK3 node.
Now, when I was reading the section on the want pathway, it honestly sounded a bit like a Shakespearean tragedy playing out at the molecular level.
The terminology is definitely evocative. The pathway begins with secreted glycoproteins called want binding to a cell's surface receptor cleverly named frizzled. But the real drama occurs deep in the cytoplasm, involving a multi-protein cluster known as the destruction complex.
The destruction complex. It sounds ominous. What proteins make up this complex?
It consists of several scaffolding proteins like axin and APC, but the critical enzymatic engine of the complex, the executioner if you will, is a kinase called glycogen synthase kinase 3, or GSK3.
Okay.
Now, to understand GSK3, you have to understand its baseline state. Most kinases we've discussed, like PKA or ACT, sit quietly inactive until a second messenger turns them on. GSK3 is the exact opposite. It is constitutively active. It is always on.
It's born angry.
Yes, and its primary job within this destruction complex is to constantly hunt down, bind to, and violently phosphorylate a specific protein called beta-catenin. But in this case, phosphorylation isn't an activation signal, it is a death sentence. By phosphorylating beta-catenin, GSK3 tags it for immediate destruction by the cell's waste disposal system, the proteasome.
Okay, let me challenge the biological logic of this, because this sounds horribly inefficient. The cell spends valuable energy and resources transcribing mRNA and translating it into this beta-catenin protein, only for GSK3 to instantly act as an executioner and chop it up the second it rolls off the assembly line. Why would evolution design a factory that instantly burns its own products?
It is a brilliant question. It looks wasteful, but it actually provides the cell with a hair trigger response system. If the cell needs beta-catenin to act suddenly, waiting to transcribe and translate it from scratch takes hours. By constantly manufacturing it and constantly destroying it, the cell creates a massive, instantly available reservoir of tension. The moment the executioner is stopped, the levels of beta-catenin explode instantaneously.
Ah, I see. It's like driving with your foot flooring the gas pedal, but your other foot is firmly standing on the brake. The engine is roaring, wasting fuel, but the moment you slip your foot off the brake, you launch forward instantly.
Exactly. And the want signal is what removes the foot from the brake. When want binds to the frizzled receptor, it activates a cytoplasmic protein called disheveled. Disheveled's job is to physically break up the destruction complex. It neutralizes the executioner. It turns off GSK3.
And because GSK3 is suddenly deactivated, the beta-catenin that the cell is constantly making is finally spared.
It is spared. It rapidly accumulates in the cytoplasm, translocates into the nucleus, and turns on a massive program of neuroprotective and neurogenic gene transcription.
And why do psychiatrists care so intensely about this dramatic little interplay? Because, as the source points out, GSK3 is directly targeted by one of our oldest drugs. We are back to lithium?
Yes, lithium again.
Earlier, we talked about lithium starving the PI cycle by blocking inositol recycling. But lithium has a second, equally important mechanism of action. It directly binds to and inhibits GSK3.
It mimics the Wnt signal. By artificially shutting down the constitutively active GSK3, lithium prevents the destruction of beta-catenin, allowing it to go to the nucleus and promote cell survival and resilience. It is fascinating that a simple elemental ion discovered to have mood-stabilizing properties over 70 years ago operates by hitting multiple, deeply fundamental nodes within the sorting room.
It provides a 1-2 punch to stabilize the manic brain. But the text adds one more layer to GSK3. It isn't just regulated by Wnt or lithium, it is also directly inhibited by ACT the kinase we just talked about in the BDNF pathway.
This is a crucial concept. GSK3 acts as a massive signaling node. It is a central point of convergence. The Wnt pathway regulates it, the BDNF-ACT pathway regulates it, GPCR pathways influence it. All these different streams of information physically crosstalk at the level of GSK3, integrating their signals to make a final, unified decision about whether the neuron should adapt, grow, or shrink.
Okay, I want to step back and look at the physical reality of the cell for a second. We have talked about CAMP floating around, IP3 releasing calcium, ROS triggering the MAPK cascade, ACT finding mTOR, GSK3 destroying beta-catenin. If you picture a cell as just a water balloon filled with a soup of thousands of different proteins, how on earth do these specific molecules find each other in milliseconds?
It's a great image, but a bit misleading.
Right, because if pKa has to randomly bump into the exact ion channel it's supposed to phosphorylate, the math doesn't work. The signal would be hopelessly slow and chaotic. How is this not just biological anarchy?
Because the cell is absolutely not a soup. It is a highly structured, meticulously organized factory floor. And it achieves this organization through scaffolding. Neurons use specific scaffolding and anchoring proteins to physically bolt related enzymes, receptors, and targets together into massive pre-assembled complexes.
The text specifically mentions AK kinase anchoring proteins. How do they work?
Think of AKPs as molecular leashes. They have one domain that tightly binds to the regulatory subunits of pKa, and another domain that physically anchors to a specific location in the cell, say, directly onto the intracellular tail of a specific calcium channel.
So pKa isn't floating around looking for a target. It is permanently bolted to the floor right next to the machine it needs to operate.
Exactly. When a GPCR activates adenylate cyclase and CAMP is produced, the CAMP doesn't have to travel far. It diffuses a few nanometers, hits the anchored pKa, releases the catalytic subunits, and the target is phosphorylated instantaneously. It ensures incredible speed and absolute specificity.
And the ultimate example of this scaffolding is the postsynaptic density, or PSD. The text mentions PDZ domains organizing the PSD.
The postsynaptic density is a massive, incredibly dense architectural structure located just under the postsynaptic membrane of excitatory synapses. It is packed with proteins that contain PDZ domains. These domains act like biological Velcro. They clip together, locking glutamate receptors, ion channels, kinases, phosphatases, and the cytoskeleton into a rigid, highly ordered signaling web.
The sorting room is meticulously organized. Everyone has an assigned desk. But what happens when this perfectly balanced, highly organized system is pushed too hard? We need to move to the next core section, clinical manifestations. We're looking specifically at desensitization, tolerance, and biased agonism.
This is where the biology directly meets the patient experience.
Let's imagine a scenario. A patient is prescribed a strong opiate for chronic pain. They're taking it every day. The drug is constantly binding to the opioid receptor, bombarding it with agonist signals. What does the sorting room do when it's overwhelmed by a single message?
This is a fundamental principle of biology, homeostasis. The cell will always fight to return to its baseline. If a GPCR is constantly activated, the cell triggers a rapid negative feedback loop called desensitization to protect itself from overstimulation. And this process is mediated by a very specific family of enzymes called G-protein-coupled receptor kinases, or GRKs.
GRKs, what is their specific trigger?
GRKs are unique because they only recognize and act upon receptors that are actively bound to an agonist. If the opiate is attached to the receptor, the GRK comes along and phosphorylates the intracellular tail of the receptor.
So it slaps a bunch of negative phosphate charges onto the inside of the receptor. What does that do?
It acts as a giant, glaring warning flag. And that flag violently attracts a protein from the cytoplasm called arrestin.
Arrestin. I assume it lives up to its name.
It absolutely does. Arrestin is a massive protein that physically binds to the phosphorylated tail of the receptor. Its sheer bulk acts as a steric shield. It physically blocks the G protein from recoupling to the receptor.
So even though the opiate molecule is still firmly attached on the outside of the cell, screaming the message, the receptor is totally silenced on the inside. The arrestin has cut the wire.
That is exactly right. This is rapid desensitization happening in minutes. But arrestin is ruthless. It does more than just block the signal. It also acts as an adapter for the cell's waste management system. It interacts with proteins called clathrin, causing the cell membrane around the silenced receptor to physically fold inward, pinch off, and drag the entire receptor deep inside the cell into a vesicle called an endosome.
The cell literally swallows its own receptor to take it off the board.
Yes. This is called receptor internalization, or endocytosis. Once inside the endosome, the receptor can either be stripped of its ligand and recycled back to the surface later when things calm down, or it can be targeted to a lysosome for permanent destruction.
Let's bring this right back to the listener in the clinic. This microscopic dance between GRK, arrestin, and clathrin is the molecular physical mechanism of drug tolerance.
Quite right.
When your patient takes that opiate daily, their GRKs and arrestins are working overtime, silencing and swallowing the mu-opioid receptors. After a few weeks, the patient comes back to you and says, "Doc, the medication isn't working anymore. The pain is back." They need a higher dose. Why? Because they physically have fewer active receptors left on the surface of their neurons. The sorting room has pulled down the mailboxes.
It's exactly what's happening.
And this is also the exact mechanism that explains the delayed downregulation of serotonin and norepinephrine receptors that we see after a few weeks of SSRI or SNRI treatment.
It perfectly illustrates how experience, in this case chronic exposure to a drug, physically alters the structural reality of the brain. But the discovery of arrestin led to an even bigger conceptual breakthrough in modern pharmacology, a concept called functional selectivity, or biased agonism.
This is one of the most exciting concepts in the textbook. For decades, classical pharmacology taught us a very simple binary. A drug is either an agonist, meaning it fits the lock and turns the receptor all the way on, or it is an antagonist, meaning it jams the lock and turns the receptor off.
Correct.
And the core assumption was that if an agonist turns the receptor on, it activates all of the receptor's intracellular pathways equally.
But that assumption turned out to be fundamentally flawed. Because, as we just established, a GPCR doesn't just have one pathway. It activates the G protein pathway, which is what we want, but it also activates the GRK arrestin pathway, which causes tolerance.
Oh, I see.
Functional selectivity is the radical discovery that the receptor isn't just an on-off switch, it is highly flexible. Certain drugs can force the receptor into a unique, twisted physical shape that highly prefers binding to one intracellular pathway over the other.
A biased agonist. It's like a key that turns the lock just precisely enough to open the deadbolt, but completely misses the mechanism for the doorknob. You get one function without the other.
Right.
And the source uses the perfect high-stakes example to illustrate this, the mu-opioid receptor.
It is the holy grail of pain medicine. Morphine is a classic, unbiased agonist at the mu-opioid receptor. When it binds, it strongly triggers the G protein pathway. This hyperpolarizes the neuron, blocking pain signals, giving the patient profound analgesia.
Right.
But, and this is the big, but morphine simultaneously strongly triggers the arrestin pathway. This leads to the receptor internalization and tolerance we just discussed. But critically, the arrestin pathway downstream of the mu-opioid receptor is also heavily implicated in the severe respiratory depression that causes fatal opiate overdoses.
So the pain relief comes from the G protein side, but the tolerance and the fatal respiratory failure come from the arrestin side.
Precisely. Researchers proved this dramatically. They genetically engineered knockout mice that physically lacked the arrestin protein. When they gave these mice morphine, the mice still experienced excellent pain relief because the G protein pathway was fully intact.
Wow.
But miraculously, they did not develop the same rapid tolerance, and they were highly resistant to respiratory depression.
Which immediately sparked a billion-dollar race in the pharmaceutical industry. The goal is to design a biased agonist opioid, a synthetic drug that binds to the mu-opioid receptor, twists it into a specific shape that exclusively activates the G protein side for pure pain relief, while completely ignoring the arrestin side, avoiding tolerance and preventing overdose deaths.
The source does note a very important caveat to this optimism, however. Even in those knockout mice without arrestin, they still developed physical dependence and withdrawal symptoms to the morphine. The biology of addiction is multifactorial and involves deeper changes in the reward circuitry, notably the dopaminergic pathways. So a biased agonist is not a magic bullet that will instantly solve the addiction crisis.
It's not a magic bullet, but it completely reimagines how we conceptualize what a drug does. We aren't just turning the receptor on, we are selectively directing traffic after it turns on. We are hacking the sorting room.
It is the epitome of precision medicine at the molecular level. And this leads us beautifully into our final major conceptual section: integration and clinical reasoning.
Let's pull it all together.
We have dissected all these disparate pieces: the CAMP, the IP3 calcium flood, the MAPK cascades, mTOR building proteins, arrestin causing tolerance. But the brain doesn't use these in isolation. We need to tie all these pathways into a unified functional outcome. And the ultimate biological example of this integration is long-term potentiation, or LTP.
LTP is the grand finale. It is the widely accepted molecular basis of learning and memory. It is the physical manifestation of how a synapse permanently changes its strength to encode a lived experience. And the classic, most heavily studied model of LTP happens at the glutamatergic synapses in the hippocampus, the memory center of the brain.
To truly understand LTP, you have to appreciate how early, rapid biochemical changes seamlessly transition into late, permanent structural changes. It is a masterpiece of cellular coordination. It involves two different types of fast, ligand-gated glutamate receptors working in a brilliant tandem, AMPA receptors and NMDA receptors.
Let's walk through it step by step.
Yeah.
Early LTP. A signal comes down the presynaptic axon. A burst of glutamate is released into the synaptic cleft. It immediately binds to the AMPA receptor, which is a simple, fast ion channel.
Right.
The AMPA channel springs open, positively charged sodium ions rush into the cell, and the postsynaptic membrane begins to depolarize. The inside of the cell becomes increasingly positive.
Now, sitting right next to the AMPA receptor in that highly organized postsynaptic density is the NMDA receptor. The glutamate in the cleft is binding to the NMDA receptor as well, but the NMDA channel refuses to open.
Why? If the key's unlocked, why won't the door open?
Because the NMDA receptor possesses a unique failsafe. At resting negative membrane potentials, the large pore of the NMDA channel is physically jammed by a single, bulky magnesium ion. It is a voltage-dependent block. No matter how much glutamate binds, nothing can pass through as long as that magnesium is lodged in the pore.
Imagine a massive, positively charged bouncer physically blocking the doorway.
That's a good visual. But here's where the synergy happens. As those AMPA receptors let more and more positively charged sodium ions flood into the cell, the local environment inside the cell becomes overwhelmingly positive.
And since, charges repel, charges.
Exactly. The accumulating positive charge inside the cell violently repels the positively charged magnesium ion, popping it out of the NMDA pore like a cork out of a champagne bottle.
The bouncer is ejected. And when that NMDA pore is finally open, it doesn't just let more sodium in. Crucially, it is highly permeable to calcium. A massive flood of calcium rushes into the dendritic spine. And we know from our earlier discussion that calcium isn't just an ion, it is an incredibly powerful, deeply impactful second messenger.
The calcium flood is the definitive trigger for LTP. The calcium immediately binds to calmodulin. That complex activates our old friends, the kinases, specifically KAMA-KI and PKC. These kinases execute early LTP in seconds. They immediately phosphorylate the existing AMPA receptors that are already in the membrane. This changes their shape, making them stay open longer and conduct even more sodium, instantly increasing the baseline efficiency of the synapse.
But they don't stop there, right?
No. KAMA-KI also mobilizes a reserve. It triggers the rapid insertion of hidden internal pools of AMPA receptors directly into the surface membrane.
So the synapse doesn't just have more efficient receptors, it physically has more of them. The next time a normal burst of glutamate is released, the postsynaptic response will be massively amplified. The connection has been potentiated.
That is early LTP. It relies entirely on modifying existing proteins. It happens in minutes, but it will eventually fade. If you want a memory to last for days or for a lifetime, you need late LTP. And late LTP requires the creation of entirely new structural proteins.
This is where all those long-distance pathways we talked about finally converge. The signal has to reach the nucleus and the local factories.
Exactly. The kinase is activated during early LTP alongside massive crosstalk from GPCR pathways like the CAMP cascade, eventually send a signal all the way to the nucleus to activate CREB. CREB begins transcribing new genes for structural proteins. Concurrently, the BDNF-TRK pathway is activating mTOR locally in the dendrite to immediately translate those new mRNAs into physical scaffolding.
It's all coming together.
The dendritic spine actually physically swells, it enlarges, it builds new scaffolding with PDZ domains to hold the new AMPA receptors in place permanently. The short-term chemical signal of a memory has literally been translated into a long-term structural adaptation of the brain's hardware.
It is truly breathtaking when you realize this exact mechanical process is happening billions of times a second in your head as you listen to this deep dive, constantly rewriting the physical architecture of who you are. And the final mind-bending point the source touches on is how modern neuroscience is learning to manipulate this internal system artificially. We are moving beyond just observing the sorting room. We're learning to remote control it. Chemogenetics.
Chemogenetics represents the absolute bleeding edge of neuroscience research. It involves using synthetic biology to engineer artificial GPCRs that do not exist in nature. The most famous of these are called DRED-Ds designer receptors, exclusively activated by designer drugs.
DRED-Ds. It genuinely sounds like science fiction. How do they work?
Researchers take a normal GPCR, say, a GQ-coupled receptor that normally responds to acetylcholine, and they genetically mutate its binding pocket. They alter it so much that it no longer responds to any natural neurotransmitter in the brain. It becomes completely deaf to the brain's internal signals.
Right.
However, they design the pocket so that it perfectly fits a completely synthetic, inert drug created in a lab, usually a molecule called CNO, or clozapine and oxide.
So they build a lock that only their synthetic key can open.
Exactly. Using viral vectors, they can insert the genetic code for these DREADDs into very specific, tiny populations of neurons in an animal, say, exclusively in the dopamine neurons of the ventral tegmental area.
Okay, let's play this out. The animal has these artificial receptors in its reward center, but the receptors are silent.
Yes, until the researcher injects the animal with the synthetic drug, CNO. The drug circulates in the blood, crosses the blood-brain barrier, completely ignores the billions of normal cells, and binds exclusively to the DREADDs. Instantly, the researcher can turn on that specific GQ pathway in that specific brain region in a completely awake, behaving animal.
They have remote control over the intraneuronal cascade.
They do. And there are also opto-XRs, which are similar artificial receptors, but instead of responding to a synthetic drug, they are activated by specific wavelengths of light delivered via implanted fiber optic cables.
By using these tools to precisely manipulate these intracellular nodes, researchers can map out exactly which behaviors, which memories, and which psychiatric symptoms are driven by which specific pathways. It proves that the future of therapeutics isn't just about indiscriminately flooding the entire brain with serotonin or dopamine and hoping for the best. It's about designing interventions that target specific nodes within the sorting room. Okay, we have covered an immense amount of ground today, from G proteins to structural neuroplasticity. Let's distill all of this down into the key takeaways from Chapter 1.11.
I would summarize them as follows. Number 1. Intraneuronal signaling involves complex intracellular cascades triggered by receptor activation. These pathways did not evolve to make things complicated. They evolved to provide massive signal amplification and allow the neuron to integrate multiple streams of information.
Number 2.
Second messengers like CAMP, IP3, and calcium, alongside their respective protein kinases, PKA, PKC, and CAMKI, are the essential workhorses that execute this signal transduction throughout the cytoplasm.
And number 3.
Neurons do not just passively transmit signals, they actively interpret them. The exact same neurotransmitter can produce wildly different effects depending on the specific receptor subtype and the current intracellular state of the sorting room.
Number 4.
These cascades are the vital bridge between short-term receptor binding and long-term adaptation. They drive gene expression via transcription factors like CREB and enable structural plasticity via local protein translation regulated by master nodes like mTOR.
Number 5.
Dysregulation at this deeply intracellular level alters how information is processed, directly contributing to the pathology of psychiatric conditions. We see this with GSK3 and bipolar disorder, or the aggressive action of arrestin driving opiate tolerance.
And then the final point, number 6.
Understanding these intracellular pathways provides the mechanistic insight necessary for robust clinical reasoning. It explains why our drugs take weeks to work, how lithium stabilizes the system, and points toward the future of drug development through concepts like biased agonism.
Brilliant synthesis. Now, let's put that knowledge to the test. It's time for our learning reinforcement segment. The expert will present 5 single best answer style questions designed to test your conceptual understanding. I will pause for a few seconds after each question to give you, the listener, time to think of the answer before the expert reveals it. Ready? Let's go.
Question 1. Which intrinsic biochemical mechanism is responsible for acting as the timer to terminate the active state of the heterotrimeric G protein alpha subunit?
Think about the built-in countdown.
Answer. Intrinsic GTPase activity, which hydrolyzes the bound GTP molecule back into GDP, causing the alpha subunit to reassociate with the beta-gamma complex.
Question 2.
In the phosphatidylinositol, or PI, pathway, what is the direct physiological consequence of IP3 production by phospholipase C?
Where did the IP3 swim to?
Answer. IP3 diffuses through the cytoplasm and binds to ligand-gated receptors on the endoplasmic reticulum, causing the rapid opening of channels and the massive release of stored intracellular calcium.
Question 3.
Which specific kinase is considered a central signaling node for regulating local protein translation within the dendrites and is heavily implicated in the rapid spine-generating antidepressant effects of ketamine?
Think of the local factory foreman.
Answer. mTOR, or the mammalian target of rapamycin.
Question 4.
What is the specific multi-step mechanism by which arrestin regulates and dampens GPCR signaling during the development of drug tolerance?
Think about how it blocks the wire and then clears the board.
Answer. Arrestin physically binds to the receptor only after it has been phosphorylated by GRKs. This binding sterically prevents the G protein from recoupling and simultaneously acts as an adapter to recruit clathrin, mediating the internalization of the receptor into an endosome.
And the final question.
During the early phases of long-term potentiation, or LTP, what specific event must occur for the NMDA receptor to finally allow its critical influx of calcium?
Remember the bouncer in the doorway?
Answer. The postsynaptic membrane must be sufficiently depolarized, typically via the heavy influx of sodium from adjacent AMP receptors, to create a positive intracellular charge that forcefully repels the voltage-dependent magnesium block out of the NMDA pore.
Excellent. If you nail those, you have a solid grasp on the architecture of the sorting room. As we wrap up today's deep dive, I want to leave you with a final thought to mull over. We spent a lot of time talking about how these internal interpreters, like the GSK3 node or the MTOR factory, can physically rebuild a synapse in response to our experiences in environment. We know that severe trauma or chronic stress can maladaptively alter these sorting rooms, changing the hardware of the brain to be perpetually hypervigilant or deeply depressed. But here's where the field of epigenetics asks a truly provocative question. To what extent are these structural intracellular changes inherited? If severe trauma permanently alters the methylation of the DNA governing the GSK3 node in a parent, altering how their cells process stress, are we passing down modified hypersensitive sorting rooms to the next generation? Are we inheriting not just the genes of our ancestors, but the biological echoes of how their neurons learn to interpret the world? It completely blurs the line between experience and biology. Something to think about. Here are the details of this episode.
Intraneuronal signaling. Byline. Beneath the signal, how neurons interpret, transform, and decide what a message means.